Welcome to today's lecture, which is the first in a two-part series dealing specifically with cell structure. What I wanted to do first was review a few concepts that we talked about previously before we continue on with today's lecture. And that concept that I want to review initially is the fact that organisms can be either single-celled, such as most prokaryotes and protists, or multicelled, such as animals, plants, and most fungi. What we are going to be dealing with in today's lecture is talking about the fundamental unit of life, which is the cell. As we all know, the basic fundamental unit of life is in fact the cell. Cells are categorized into two major groups. Cells can either be considered prokaryotic or eukaryotic, and we will review those two broad categories of cell types in just a minute. Prokaryotic cells are single-celled organisms and are comprised of the domains bacteria and archaea. Eukaryotic cells comprise the domain eukarya and can be either single-celled or multicellular organisms. These are cell types that we know as protists, plants, fungi, and animals. But before we discuss those two broad categories of cell types, I want to refresh our memories with a concept that I mentioned previously, and that is every single cell, no matter what cell type it is, has some shared characteristics. Every single cell has a thin plasma membrane, which is the boundary of the cell. Now, the tail end of Unit 1 discussed plasma membrane structure, and our last lecture, the first one in this particular unit, talked about transport across that membrane. In addition, every single cell will have DNA at some point in its life cycle. DNA is the genetic material. It contains all of the instructions necessary for a cell to survive. So every single cell, no matter if it's prokaryotic or eukaryotic, will also have DNA at some point in its life cycle. And then the last shared characteristic of every single cell is a structure known as the ribosome. And we'll talk about ribosomes in a little more detail in today's lecture. But ribosomes are essentially protein synthesizing machines. They are structures within the cell that synthesize proteins that the cell needs to survive. One last term that I want to mention at this particular point is the term cytoplasm. This may be something you've heard about previously. Cytoplasm is defined as the entire contents of the cell. So everything inside the cell is considered the cytoplasm. Let's go ahead and take a closer look at prokaryotic cells first. Prokaryotes are typically smaller than eukaryotic cells, and they lack internal structures that are surrounded by membranes. And we'll talk about those structures when we talk about eukaryotes in general on the next slide. As I mentioned just a minute ago, these particular cells lack a nucleus, and a nucleus is a membrane-bound structure that houses DNA. Most prokaryotic cells will also have a rigid cell wall, and this allows for structure and protection of the prokaryotic cell. Now, we won't talk about prokaryotic cells in much more detail than this one particular slide in this class. You could take a microbiology class where the focus would definitely be more on the prokaryotic cell type. So for now, let's talk about the things that are common in a typical prokaryotic cell. As I mentioned on the last slide, every single cell is going to have a plasma membrane or a cell membrane, and this is a membrane that encloses the cytoplasm and acts as the boundary or the border, dividing the interior environment from the exterior environment. As I mentioned on the last slide, every single cell type will also have ribosomes, which are the structures that synthesize proteins necessary to carry out life. I also mentioned on the last slide that every single cell will have DNA at some point in its life cycle. Prokaryotic cells do in fact have DNA. They do not have that specialized structure known as the nucleus to house the DNA, but they do have a region of the cell where the DNA is found, and this is referred to as the nucleoid region. In addition, like I mentioned just a minute ago, prokaryotic cells will have a cell wall, which is a rigid structure outside of the plasma membrane that provides structure and, more importantly, support to these single-celled organisms. Now, some prokaryotic cells may also have something known as a flagella, which is plural, and flagellum, which is singular. And this is a locomotor appendage, and that's what you see in this figure that looks sort of like a long tail protruding from the prokaryotic cell. The flagellum, again singular, is a locomotor appendage, and this allows for the prokaryotic cell to move in its environment. Some prokaryotic cells will also possess a capsule, which is an outer polysaccharide coating. This usually is a defense component that prevents engulfing of the prokaryotic cell. So other organisms can't necessarily attach or attack this cell if it has an exterior capsule. Lastly, not all, but some prokaryotic cells will have pili. And pili is plural, pilus is singular, and that in this figure looks like those little hairs that are sticking off of the prokaryotic cell. These are structures that are oftentimes used for attachment, either attachment to other cells or attachment to surfaces in their environment. The other broad category of cell type is a eukaryotic cell. And when we talk about eukaryotic cells, we oftentimes subdivide them into an animal cell and a plant cell. And these, again, are generalized figures that we're looking at on this particular slide. However, eukaryotic cells are fundamentally similar to one another. The most important defining feature or characteristic of a eukaryotic cell is in this figure, that purple structure, that is the nucleus. The nucleus is a membrane-bound structure, meaning it has its own membrane associated with it, and this is where the DNA is found or housed, and it allows for protection of that genetic material. In addition to that, eukaryotic cells also have organelles. Organelles are structures with specialized function, and we are going to talk extensively about those in our next lecture. The first organelle we're going to focus on is the nucleus. The nucleus is the defining characteristic that differentiates prokaryotic cells from eukaryotic cells. Prokaryotic cells lack a nucleus, whereas eukaryotic cells do in fact possess a nucleus. The nucleus is the organelle that houses the genetic material or the DNA of the cell. Within the nucleus, you will find chromatin, which is a complex of DNA and proteins, and I'm going to come back to that in the next slide. For now, I want to talk a little bit more about the nuclear structure. Within the nucleus, you will find the nucleolus, and the nucleolus, again, is sort of at the center of the nucleus, and it is a specialized structure that is the site of ribosome synthesis. Ribosomes, which we discussed previously, are protein synthesizing organelles, but there is in fact a location that those ribosomes themselves are produced or synthesized, and that occurs in the nucleolus of the nucleus. I would like to now go ahead and review the structure overall of the nucleus. It does in fact have a membrane, however it has a double phospholipid bilayer. Basically, you have a phospholipid bilayer like the typical membrane structure we've discussed previously, that is folded on itself again to form a double phospholipid bilayer. The nuclear membrane itself is referred to as the nuclear envelope, and the nuclear envelope has openings in it that are referred to as nuclear pores. Essentially what this looks like is a wiffle ball. If you can imagine what a classic wiffle ball structure looks like, it's a ball with a bunch of holes in it. Essentially that's what the nucleus looks like. It is a structure, again, that houses the genetic material, but has a lot of these openings, these nuclear pores, that allow for materials to enter, but more importantly, to exit the nucleus. And this will become important when we start discussing how genes are expressed and the functional products associated with genes. In addition, on the inside surface of the nuclear envelope is something known as the nuclear lamina. It is a net-like array of protein filaments that line the interior surface of the nucleus and help maintain shape and provide structure and rigidity for the nucleus. And this makes a lot of sense because the job of the nucleus is to protect and house the DNA. And it makes sense that this is fortified or strengthened to make sure that that genetic material is in fact safe. Now, as I mentioned on the previous slide, the DNA found inside the nucleus of a eukaryotic cell is not what we would consider just naked DNA. In this particular figure up at the top, you see the classic helical shape for DNA. But in eukaryotes, the DNA is found in complex association with proteins. These proteins are known as histones, and it is the complex of DNA and proteins that is then referred to as chromatin. Chromatin is further condensed into the classic characteristic chromosome structure, and chromosomes are really just specific structures that the DNA is found in. The ribosome is the next organelle that I want to discuss. Ribosomes are structures that synthesize the cell's proteins. They are complex structures that are made up of a molecule known as ribosomal RNA, as well as ribosomal proteins. In fact, it is such a complex structure that it's subdivided into two subunits, something referred to as the small subunit and the large subunit. Now, when we get to Unit 4, we will talk about ribosomes again in much more detail and their role in protein synthesis. But for now, what I do want to talk about just a little bit is the fact that ribosomes can be found in two different locations within the cell. Some ribosomes are referred to as being free, meaning that they are not attached to any specialized structure inside the cell. And these ribosomes synthesize proteins that are generally needed for the cell to survive. In addition, ribosomes can be what are referred to as being membrane-bound or membrane-associated. And that means that they are specifically bound to a membrane inside of the cell. That cell component that they are bound to is referred to as the Endoplasmic Reticulum, and we're going to talk about that in a little more detail in just a minute. But what I want to highlight in the right-hand figure is those ribosomes that are bound to the Endoplasmic Reticulum, also referred to as the ER. Those ribosomes become attached to the ER membrane, and the proteins that are synthesized get threaded into a space known as the lumen of the ER. These proteins typically get modified and are proteins that are either cell membrane proteins or proteins that are going to be exported and secreted from the cell. So free ribosomes synthesize proteins that are utilized inside the cell, and typically the membrane-associated ones, the ones that are bound to the ER membrane, are going to synthesize proteins that are either membrane-associated proteins or proteins that are going to be exported or secreted from the cell. There is, in fact, a relationship between the two organelles that we just discussed. The nucleus, which we talked about, houses the genetic material, or the DNA, and the ribosome, which is involved in protein synthesis. What you see in this particular slide is a summary of what happens for or during protein synthesis process. The DNA houses the genetic material. It contains all of the information or the instructions to produce the proteins that the cell needs to survive. The DNA is copied in a gene-by-gene basis in a mechanism known as transcription to produce a molecule known as RNA, more specifically mRNA. mRNA is a copy of one gene and is the instructions typically to synthesize a specific protein. The mRNA copy needs to exit the nucleus and enter into the cytoplasm where the ribosomes are found. This is why those extensive amounts of pores that we discussed in the nuclear envelope are important for the export of mRNAs from the nucleus to the cytoplasm where the ribosomes are found. The mRNA, once it exits the nucleus, encounters ribosomes, and this is where the instructions are read and proteins are synthesized. We will discuss this again in our fourth unit of material, but I just wanted to tie together those two structures for you. Before we continue discussing organelles in the eukaryotic cell, I want to touch on the concept of the endomembrane system. Now, the endomembrane system is a system within the cell of membranous organelles that either have a direct physical contact via membrane or they are in contact with one another by vesicles. Vesicles are small membranous sacs, and we'll look at those in a little more detail in just a minute. But the endomembrane system is a network or a system within the cell where, again, there is either direct physical contact of membranes from one component to another, or there's interaction via membranous sacs known as vesicles. This is a great illustration that includes components and organelles that we have not yet discussed, but we will, and so I'll come back to this particular figure so that we can review the organelles once we've talked about them. But because I just introduced this endomembrane system, what I wanted to do is give you a little bit better feel for what's happening in the endomembrane system. Again, you either have direct physical contact of membrane, and we'll look at some examples of that coming up in the next slide or so, or you have interactions via vesicles that are membranous sacs that allow for transport of components from one organelle to another in the endomembrane system. And so, again, it is a very complex network of membranous organelles where the membranes contact with one another or they are in contact via transport vesicles. The next organelle I want to discuss is the endoplasmic reticulum, also known as the ER. Now, I mentioned the ER briefly when discussing ribosomes, and that ribosomes could either be found free in the cytoplasm of the cell or the cytosol of the cell, or they could be bound to structures known as the endoplasmic reticulum. So now I want to talk about the ER in a little more detail. It is a very complex structure that is subdivided typically into two broad categories known as the smooth ER and the rough ER. Now, the ER itself is an extensive membrane-bound network that is continuous with the nuclear envelope. So, as you can see in this particular figure, in the center of the figure, you can see a portion of the nuclear envelope, and then in the darker purple color is the ER membrane, the membrane of the ER. And here you can see that there is a direct physical connection from the nuclear envelope to the ER membrane. When describing the structure of the ER, what you can see is what it looks like is sort of a network of tunnels that connect directly from the nuclear envelope. The network of tubes and sacs are known as cisternae, which is plural, or cisterna, which is singular. Within the cisternae is the cisternal space, and this is the internal compartment within the ER. And this holds true for both smooth ER and for rough ER. We're going to look in the next slide in a little more detail of what's happening in that cisternal space. But for now, I just want to make sure that we orient ourselves to the structure overall of the ER. As I mentioned at the beginning of the slide, The ER is divided into two categories, the smooth ER and the rough ER, and these components or subcategories of ER got their name because of the way they looked under the light microscope. The smooth ER had a very smooth appearance to it, and the rough ER seemed to have little bumps all over it, making it look rough in appearance. We now know that what makes the rough ER rough is the fact that ribosomes are bound to the ER membrane, and that's what makes it look like it's rough. Again, we'll look at that in a little more detail in the next slide. But there is a differentiation with what the ER is doing when we're talking about the smooth ER versus the rough ER. So let's talk about the smooth ER first. The smooth ER, again, is smooth in appearance, does not have ribosomes bound to its membrane, and is involved in lots of different synthetic processes. It's involved in synthesis of lipids, more specifically phospholipids and steroids. In animal cells, this particular component is involved in glycogen metabolism. Glycogen is the stored structure for energy, and it is basically made up of repeating glucose molecules. And so this can be metabolized or broken down into individual glucose molecules for the cell or the organism to utilize. The smooth ER may also be involved in detoxification, things like drugs and poisons within the liver of an animal system. Hydroxyl groups can be added on to these drugs or poisons, making them much more soluble and allowing them to be flushed from the system. Additionally, the smooth ER can be involved in calcium fluxes. Differences in concentration of calcium oftentimes acts as what's known as a secondary messenger inside of the cell. These changes in calcium concentration send signals to different parts of the cell, and the smooth ER can be involved in allowing for these calcium fluxes to take place. And as I mentioned previously, the rough ER gets its name because it looks rough in appearance under the light microscope, and it looks rough in appearance because ribosomes are physically attached to that ER membrane, or to that portion of the ER membrane. Specifically, what happens in this particular location is the synthesis of secretory proteins, that is, proteins that are going to be secreted from the cell, and membrane-bound proteins. And we're going to look at that in a little more detail on the next slide. So this is a zoomed-in view of the rough ER itself. What you can see is the phospholipid bilayer that is part of the ER membrane. You can see it sort of looping around to create this space inside the cisternae, or the cisternal space. You can also see in blue the ribosome that is attached to this membrane. So again, what this does is highlight two characteristics we've talked about: ribosomes, which are involved in protein synthesis, and the ER, specifically the rough ER. What happens here is ribosomes attach to the ER membrane, and as they are synthesizing proteins, those proteins get threaded into the cisternal space. Like we discussed previously, the initial synthesis of a molecule in this case is known as a polypeptide, and it isn't until there is folding into a final functional conformation when we can change the terminology to a protein. But what happens in this particular case, again, is the ribosome is involved in the synthesis of the polypeptide leading to protein. That protein is threaded and fits into the lumen, or the space, of the ER. And the types of proteins that are synthesized here are either going to be proteins that are going to be secreted from the cell, or they're proteins that are integral parts of membranes. In this figure, this happens to be a protein that is going to be secreted from the cell. What happens is the protein gets packaged in membrane, that's part of the ER, and this vesicle basically buds off the ER and is then directed to eventually reach the plasma membrane, where fusion can occur and release can take place. what you should be able to do is recognize that this would be a mechanism of exocytosis. In discussing how large molecules enter and exit the cell, we discussed the fact that large molecules can enter the cell via endocytosis and exit the cell via exocytosis. So this would be where this protein is going to be secreted from the cell. It would have to then interact, the vesicle would have to interact and fuse with the plasma membrane to release the contents. The next organelle we're going to focus on is known as the Golgi Apparatus. It too is made up of a phospholipid bilayer that has been sort of folded back up on itself, so each compartment of the Golgi apparatus also has a lumen or a space inside it. It kind of to me looks sort of like a stack of pancakes, one right on top of the other, and what you can see in the cross-section view in both of the images on the left-hand side and on the right-hand side is that sort of stack of pancakes look overall. Each one of the compartments is connected to the other by vesicles, and how molecules get to the Golgi are also by vesicles that are transported from one location to the other. So this is still part of the endomembrane system. So let's talk a little bit about what this particular organelle does. This organelle sort of is like a jack of all trades. It has lots of different functions associated with it. It is a site of manufacturing, that is, synthesis of polysaccharides that get incorporated into cell walls can occur in the Golgi. It can act like a warehouse, basically to store molecules for certain periods of time. It acts as a sorting center, so molecules that get targeted to the Golgi then redirects them to where they need to go within the cell. It can also be a site for modification of proteins. For example, glycoproteins are a category of proteins that have carbohydrates covalently linked to them. This is where that modification would take place. Lastly, it also acts as a shipping center. Molecules get in, as we said, get sorted out, and then get shipped to their final location within the cell. So you can see it sort of acts in lots of different functions within the cell. What I also want to highlight from this figure on the left-hand side is you can see a vesicle in the purple color coming towards the Golgi, and then you can see a vesicle in the bottom right-hand corner of the figure leaving the Golgi. The receiving end is referred to as the cis face of the Golgi, and the opposite end where the molecules are leaving within vesicles is referred to as the trans face of the Golgi. The next organelle that is still part of the endomembrane system is referred to as a lysosome. The lysosome is a membrane-bound structure that contains digestive enzymes, also referred to as hydrolytic enzymes. These are molecules that speed up the breakdown of macromolecules. These organelles also can play a role in breakdown of damaged organelles, so organelles that are no longer functioning properly. Components get recycled, and the way that they get recycled is to be broken down into individual components that can be then utilized in other ways. Again, these are membrane-bound organelles. They are part of the endomembrane system. And I want to go back to the term hydrolytic enzyme. The word hydro refers to water, and lytic has the word or root lys in it, as does the term lysosome. Lys means to break apart. So hydrolytic enzymes are enzymes that use water to break apart molecules. And when we discussed macromolecules back in Unit 1, we discussed the common theme, or the way macromolecules are broken down, are by hydrolysis reactions. Hydrolytic enzymes would be those types of enzymes that help in hydrolysis reactions. So the term lysosome, like I just mentioned, has the root lys, which means to break apart. And again, this structure contains enzymes that are going to help break apart molecules into smaller, simpler components. The last organelle I want to discuss in today's lecture, which is still, again, part 1 of our cell structure lecture, and contains an organelle that is still part of the endomembrane system. So all of the organelles that we've discussed in today's lecture are in fact part of this endomembrane system. Again, a system of organelles that are connected either physically by membrane or come in contact with one another by membranous vesicles that carry contents from one component to the other. So again, the last structure then that I want to discuss that's part of the endomembrane system are vacuoles. Vacuoles are large membranous sacs or membrane-bound sacs. Think of them as a larger, bigger size vesicle. So vesicles are small membranous sacs. Vacuoles are large membrane-bound sacs. There are, in fact, lots of different categories or subcategories of vacuoles. You can have something that's referred to as a food vacuole, which would be a structure that would contain molecules that were taken in as part of a nutrient acquisition mechanism that you can see in the bottom figure in the slide. In the top left figure, you can see something referred to as a contractile vacuole. Hopefully what you can see between those two figures in the left-hand corner is the vacuole closed versus open. This is a specialized structure involved in osmoregulation. So for this particular cell type, it's important to be able to regulate the amount of water that is in the cell, and the contractile vacuole allows for opening and closing to allow for water to enter or exit into this particular type of cell. More commonly, in the right-hand figure, you can see a structure that is found in plant cells. This is referred to as the central vacuole, and it is a fluid-filled vacuole that typically acts as a reservoir for organic compounds, ions, food, and waste.